Hello friends, today we are going to study about protein secondary structure. Till now we studied about protein primary structure which includes amino acid sequence and composition. In the protein secondary structure we are going to study about alpha helix, beta sheets and turns. So what do we mean by secondary structure of a protein? Secondary structure means it is the local arrangement of a polypeptide backbone. The two conformations are mostly found in the protein, that is alpha helix and beta conformation, which constitute the secondary structure of the protein. And these secondary structures has been predicted by Pauling and Corey in 1951. The first is alpha helix. As the name suggests, alpha helix means helical turn. So alpha helix is the helical arrangement of polypeptide backbone against an imaginary longitudinal axis. And in these arrangement, the amino acids that form these helical structure, their R group or the side chain that protrudes outside the helical structure. So alpha helix, these are mainly stabilized by the hydrogen bonding. And this bond is present between the amino acid present at nth position with the amino acid present at n plus fourth position. So with every fourth amino acid, the hydrogen bond is formed. If we see these turns, so these turns are stabilized by 3 to 4 hydrogen bonds. So the alpha helix is the arrangement of the polypeptide backbone, which is mainly stabilized by hydrogen bonds. This alpha helix conformation, they range with phi and psi value of minus 60 degree and with psi value of minus 45 degree to minus 50 degree. 1/4 of all amino acid residues in any polypeptides, they are found in alpha helix form. Now, if we see this helical turns, these are right-handed turns, and each turn consists of 3.6 amino acid residues. Pitch is 5.4 angstrom. What do we mean by pitch? This height. This is 5.4 angstrom. Average length of this alpha helix is, it consists of approximately of 12 residues, resulted in 3 helical turns, and the total length will be 18 angstrom. The distance between nth and n plus 4 residue is 2.8 angstrom. That means this distance is 2.8 angstrom where hydrogen bond is formed. The core is stabilized by van der Waals contact. Now, why does the alpha helix form more readily than any other possible conformation? Because it utilizes the maximum potential of hydrogen bond. So, why the alpha helix is more readily formed? Because they do the optimal use of internal hydrogen bonds. Now, each successive turns of alpha helix is stabilized by 3 to 4 hydrogen bonds. What is the amino acid sequence that can affect the helix stability? Now, all the polypeptides cannot form stable alpha helix. To form a stable alpha helix, the sequence of amino acid is very important. Why so? What will happen if the polypeptide contain a long stretch of glutamate residue? What will happen in that case? Glutamate is a negatively charged amino acid. If the glutamate residue is present at a long stretch, what can happen? The negative negative will repel each other and it cannot form a stable alpha helix. Similar with the case of lysine or arginine residue. A long stretch of lysine and arginine in a continuation cannot form a stable alpha helix because these are all positively charged. So the positive positive will repel each other. What will happen if it contains a long stretch of asparagine, serine, threonine and cysteine residue? These are all bulky. They will create steric hindrance. What will happen if it contains a stretch of proline and glycine? Proline has a unique structure which gives more rigidity. So if a stretch of proline is present in an alpha helix, it is difficult for alpha helix to bend and take any turn. So a stretch of proline is rarely found in an alpha helix. What will happen if a stretch of glycine is found? It will make the alpha helix too flexible. So the sequence of amino acid is very important for the stability of alpha helix. These charged amino acid, they are present if in the nth position any negative charge amino acid is is present, in the N plus fourth position, a positive charge amino acid will be found. These two then can take part in electrostatic interaction. Similarly, if in the nth position any hydrophobic amino acid is found, then in the nth plus 4 position also a hydrophobic amino acid will be found. This will take part in hydrophobic interaction. Similarly, aromatic amino acids are also spaced in such differences. So a long stretch of any charged or a bulky or an aromatic amino acid can affect the helix stability. All these amino acids should be spaced equally, and that distance is 3 to 4 amino acid gap. Here the same thing is written. The positively charged amino acid are often found 3 residue away from negatively charged amino acid. That helps in the formation of electrostatic interaction. And similarly, aromatic amino acids are also equally spaced and they also participate in hydrophobic interaction. Next is, because an amino acid or a polypeptide have an N-terminal and a C-terminal end. N-terminal end means amino terminal. Amino terminal means positively charged. Similarly, C-terminal is carboxyl terminal, means it is negatively charged. So the polypeptide along the axis, it has slightly of dipole nature. It carries some charge. The oxygen end is having the negative charge and the hydrogen end will have a positive charge. So in an alpha helix, the end amino acids also affect the stability of the conformation. Towards the negative end, the ends should contain the positively charged amino acid. And towards the positive end, the ends should contain negatively charged amino acid. So, in a polypeptide, because it has an N terminal and a C terminal, it has a slight dipole character. Because of this dipole character, the ends of the alpha helix is very important for its stability. Towards the negative dipole, positively charged amino acid should be present. And towards the positive dipole, the negatively charged amino acid should be present. Now, what are the 5 constants that affects the stability of an helix? First is electrostatic repulsion or attraction. The successive amino acid should not be continuously positive or negative, they will repel each other. A continuous stretch of bulky amino acids also cannot present in alpha helical structure because it will disturb the helical conformation because of steric hindrance. The interaction between the N and N plus 4th amino acid is very important in maintaining the stability. If they are not properly spaced, they cannot help to maintain the conformation of alpha helix. The occurrence of proline and glycine. Proline is rarely found in alpha helical structure because it provides rigidity. Glycine is too flexible. And next is the ends of the alpha helical structure. Because of innate electric dipole, the negative end should have positively charged amino acid and positive end should have negatively charged amino acid. Otherwise, it will affect the stability of an alpha helix. Next is beta sheets. It is of a zigzag conformation. The beta sheets are also stabilized by hydrogen bonding between the neighboring polypeptide chains. The chains in the beta sheets, these are arranged like this. Okay, and the hydrogen bond is present between the adjacent chains. The beta sheets can be found in the parallel and antiparallel form. The antiparallel beta sheets, the hydrogen bond formation is proper. As you can see, they are formed in a proper way, in a proper orientation. But in case of parallel beta sheets, the hydrogen bond formation is in a zigzag pattern. Therefore, antiparallel beta sheet is more stable compared to parallel beta sheets. What do we mean by antiparallel or parallel beta sheets? If the arrangement is like this, that means amino terminal of one chain is in front of carboxyl terminal of another chain, then it is known as antiparallel. If the amino terminal of one chain is in front of amino terminal of another chain, then it is known as parallel beta sheets. In case of antiparallel beta sheets, the hydrogen bond can be formed in a proper way, hence it is more stable compared to the parallel one. Now, due to pleated edge-like appearance, the beta sheets are also known as pleated sheets. If you can see the structure, helical looks like this, and the beta pleated sheet is like a pleated sheet arrangement. Now, beta sheet, the length is approximately of 2 to 22 strands and average is 6 strands. Its length is of approximately 15 residues. Parallel beta sheets are less stable compared to antiparallel, and they also have a right-handed twist. When two or more beta sheets are layered close together, their R groups should be small. If they're very close, they should have a very small R groups, otherwise they will create again the steric hindrance. So the two amino acids that have the smallest R group are glycine and alanine. Therefore, in beta pleated sheets, we can find more of glycine and alanine amino acid residues. Let's come to the turns. Turns means when a beta sheet is present or a helical is present, the two structures are connected with the help of turns. The turn consists of 4 amino acid residue. There is a hydrogen bond between the first and the fourth amino acid residue. The second and third amino acid residue, these are involved in hydrogen bond formation with water molecules. Turns are the connecting links between two secondary structures, either two beta sheets or two alpha helix or beta sheets with alpha helix. So this is the connecting link. The turns mainly consist of glycine and proline. Why glycine? Because it provides flexibility. Why proline? Because it will help in getting a little of rigidity. These are nearly found at the surface of the protein, and the central two amino acids participate in hydrogen bond with water molecule. Turns are of two types, type 1 and type 2 turns. These two turns only differ here, if you can see, in the orientation. The most common turn is the beta turn that connects to antiparallel beta sheet, and the rarest turn is gamma turn, which consists of 3 residues. The beta turn consists of 4 amino acid residues, whereas gamma turn consists of 3 amino acid residues, but this is rare. The most common is the beta turn. Now, one question from my end for you all, that is, why would you be unlikely to see an helix containing only the following amino acids? That is arginine, lysine, methionine, phenylalanine, tryptophan, tyrosine, and valine. So you have to tell me that why a continuous stretch of these amino acids is not found in an alpha helical structure. Do let me know in the comment section. If you found the video informative, please do like, share, and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.